Hello, everyone, and welcome to the Dendrite Podcast. My name is Farnam, and I'm here with my co-host Saeed. In the Dendrite Podcast, we try to help students explore ongoing research and get involved in possible opportunities.
This is a conversation with Dr. Richard Naud. He's an associate professor in the Department of Cellular and Molecular Medicine at the University of Ottawa and a scientist in the Neuroscience Program at the Ottawa Hospital Research Institute. He started by studying physics at McGill University. During his doctoral studies at EPFL, he designed statistical methods for an automatic characterization of neuronal dynamics. He also co-organized a spike timing prediction challenge for the comparison and benchmarking of mathematical neural models.
After obtaining his Ph.D., he developed quasi-renewal theory, which allows neural responses to be decoded in the presence of adaptation. He obtained different post-doctoral scholarships at uOttawa, Cambridge University, and Technical University of Berlin, identifying computational roles of active dendrites and inhibition in sensory systems and neocortex. We hope you enjoyed this episode, and now, without further ado, let's say hi to Dr. Naud. Hello, Dr. Naud. How you doing?
Hello. Very good. Happy to be here.
Awesome. Well, we really appreciate your coming to our podcast, and we're really grateful. Let me just start by saying, how's the sabbatical going? I know you've been in France for a while now, and just tell us maybe a little bit more. How's it going, and how different is it from Ottawa?
Well, I feel I'm pretty lucky to be in the sabbatical right now. I feel I have time to do what I feel researchers are kind of supposed to do. I have time to read papers, do some theory, write down equations, discuss with other scientists, and really do what I think research is all about. So I'm pretty happy about that.
Well, are you there just by yourself?
I'm here with my family, so the whole
Okay.
family. So my kids are actually going to school in France. They're learning proper French. I mean, we speak French at home, but their French has improved dramatically since we got here. And yeah, my wife is here too. But I have friends as well at the University of Bordeaux, where I'm interacting with scientists.
Cool. That is great.
I'm also using this as an opportunity to meet friends from Europe. So I'm going to give talks here and there. Next week I'm in Oxford. A few weeks after that, I'll be in Berlin. So yeah.
Oh yeah, that would definitely be great.
It's a great chance having the whole Europe just like one train away. So
That's right.
all right, so maybe you can start by asking you about all the different institutes that you have been throughout your education, just academic journey from McGill to you've been to quite different countries and very, very impressive institutes, EPFL, Berlin, Cambridge, and I know that you also have a lot of collaborations. I think I saw MIT somewhere, and now, as you said, like even in the sabbatical, you're doing talks in Cambridge, in Oxford. So, how does this diversity and this just whole array of different institutes help in maybe shaping your way of understanding and the way of thinking, or how does it affect your scientific interest?
Well, I started with the PhD in Switzerland. You mentioned EPFL. I decided to do that PhD in Switzerland, despite all my friends at the time at McGill going to the US. I didn't want to go to the US, and I was looking into Europe, mainly because I was kind of attracted by the way of life. But then I realized there are major advantages to do a PhD in Europe, being that you get a pretty decent salary as a PhD student, and then scientifically, at least in Switzerland, science is really excellent, and you have the opportunity to connect with a lot of people because all these different centers are close by, as you were mentioning earlier. So during my PhD, I gave, something like 20 talks in different countries. And it's not because I was any sort of big star. It's more because, regular meetings, they're fairly small meetings, but they were always in different countries. And I met with a lot of different researchers from different places. And that allowed me to get a much broader picture of what science was about, at least in Europe. but also to connect with a number of scientists that share my point of view or share the excitement about a sub part of science. And these are people that are still talked to. So even
Mhm.
like I'm in Canada, I have regular meetings with people in Switzerland, in Germany, that are professors now, but we share a research interest and we talk every 2 weeks or every month. And a lot of insights come from that.
Cool. That is amazing. I'm sure there's tons of connections that you can make outside of Ottawa. And I really look forward even for that opportunity, even myself, to be able to like go out there and see what other people might thinking about neuroscience. Let me just ask you about, so you had so many interests throughout your whole journey and stuff, but I'm just wondering, how does physics help you a lot? Because mostly the science students are not that strong in math or physics, but I'm sure when you're talking about neuroscience, physics and maths play a key role. So I'm just
Yeah.
wondering how that affect you along the way, and what
Mhm.
could you tell the science students who are interested in neuroscience how they should improve their other aspects of science, which is the physics and
Mhm.
math?
Yeah. Yeah, that's a very important question. So in my lab, I've seen now what different backgrounds mean in terms of how you approach different problems or how you tend to approach different problems. And I mean, I'll bring in a few stereotypes. These are stereotypes that they're not meant to mean that they represent everyone. Everyone's very different, but certainly a tendency that I've seen in general. And I take people from biomedical background, from computer science, from math, from physics. And these different backgrounds um, will give the students some aptitude, and it's basically strengths and weaknesses. I'm a physicist, I came from physics. Um, and the big aptitude there is that in physics, we're trained to translate a physical system into a set of equations, and to basically the set of equations is just there as a way to have a symbol and help us ground ourselves, but really the goal is to try to put ourselves within the system, to try to visualize what's happening physically and have a really precise visual picture of what's happening. And when you're able to do that, you're able to predict what will happen in different scenarios, and you're able to generalize. So that's the nature of having an understanding, and that's a very rare and difficult uh, tool to have. It is very efficient in neuroscience because sometimes I have discussions with experimentalists, for instance, and they're like, "Oh, we don't know whether this is going to lead to this," and I'm like, "No, we know." you you have to put yourself into this and then argue or explain like this leads to that and this leads to that. All these sequence of logical steps have been done through the integration of the mathematical concepts. However, these are the strengths. There are also weaknesses, and physicists tend to oversimplify things. There is a tendency of, in order to deeply understand something, physicists tend to always start with the simplest scenario and then build from there. And there's, alongside this, a unwillingness to tackle the diversity or the complexity that is particularly important in neuroscience. And so there's kind of a closed-mindedness that is not very conductive in neuroscience. You do need to navigate the complexity. And that's where like biomedical students in a position are really good at. So people in biomedical training tend to navigate the complexity of things very well. They don't mind looking at a subject and realizing there's so many different exceptions and memorizing all the exceptions in all the different cases. Um, and that's very useful, especially when you combine that with a training that tries to summarize and bring everything under one hood. That is what we do in my lab. So people that come in my lab, if they have a physics background, I'll have to train them to open their minds to the complexity and to navigate the complexity of neuroscience, how you go about doing this, and so on. People that come with a biomedical background, I'll have to train them to go and try to put themselves in the systems and understand the equations in order to have a way to generalize. And that's something, actually, I should say, is a bit of an anecdote, but you might like. At the very beginning, I was a little bit reluctant in taking biomedical students. I didn't know whether this was working, and people have been saying to me, it doesn't work this way around because it takes so much time to learn all the math. And math is very like sequential. You can't like learn it in parallel. You have to go one thing after the other. And so it's too long to wait for a student to get there. Whereas if you go from physics, you can learn the right element in neuroscience and you're good. And I've had like 2 or 3 maybe exceptional students from biomedical sciences that were really, really interested in getting into computational stuff. And they were ready to do, of course, it's added work. You need to complete your training with math, and it's not easy. And it's not necessarily the first thing you will do. But if you're ready and you're really interested in doing this, they were able to do this. And then they came out with this combining of the strength of the two sides and had very powerful results as a consequence.
Right.
So, a student that's now in like one of the most prestigious institutes, just graduated, my first PhD student, came from biomedical sciences and had an exceptional PhD.
That is amazing. Yeah, I'm sure we definitely need more work on kind of combining all of these things together as in like an undergrad. Because like as a biomed student, I could say most of our class are more about biology, and we never really, even if you're specializing in like neuroscience option in biomed, you don't really get a chance to like learn about like coding and the more physics aspects of neuroscience. So hopefully that's something we can definitely work on in the near future. Let me just now start by saying that what are you doing in your lab? How's it going in your lab? And for the audience who are interested in our lab, just wondering what is it that actually happening right now in Doctor's Not lab?
You mean research-wise?
Yeah.
Yeah. There's 3 main research axes that we do in the lab. Right now, at the moment, there's 3 main axes. One is connected to the dendrites. I spent a bunch of my time working on dendritic computation, understanding what dendrites add to the signal processing power of neurons. And one type of computation I've focused on was its role in learning. So one axis is a theory of learning. So how neural network organize themselves and coordinate learning among many, many different neurons so that they can learn something that's meaningful while still like learning with independent processes. The other axis is biophysical mechanism of microstimulation. So microstimulation has been a very, very old technique, putting an electrode in the brain of someone or an animal and stimulating neurons in this way. But we don't really know what that does to the nervous tissue. There's some papers on this, but there's big gaps. And so we're working on understanding the biophysical mechanisms of microstimulation in cortex, and it involves really surprising aspects of neural dynamics. And the last axis is that of reaching a theory for serotonin with my collaborator, Jean-Claude Beig. We've been working a lot on the serotonergic system. And in my lab, we're building a theory of what serotonin tells the brain.
Interesting. That's very cool.
That is really interesting. So all the 3 ones. So if we were to ask a question about the first one, you said that you're trying to develop a theory about learning. How does that theory or your attempt talk to maybe other theories about learning, mainly in the cognitive sciences or psychology? So are you trying to make any connection with that, or no, it's a completely novel approach?
Yes, no, very much so. The way I see a lot of what computational neuroscience has to do is we're trying to, everybody in neuroscience tries to understand the relationship between neural data and function.
Mhm.
We have neural data, we see things, we see synapses, we see neurons responding, we see fMRI hotspots, and we try to relate this data with the function, right? But the two world, like the connection seems pretty straightforward when you put it this way. When we start doing this, you have like, millions of neurons firing at the same time, changing synaptic strength at the same time. How can we relate that to function like learning? The two worlds are very far. It's not easy to see how we can connect the two. And so the field, or at least in my lab, what we try to do is we have like a 4-step approach. From the neural data, we can build mathematical neural models. These mathematical models give encapsulate the data in the mathematical terms that we can study on their own and study the properties and explore what they can do, the total extent of what they can do. On the other hand, if you think of functions, the functions typically like learning in psychology might be a little bit loose. You use the word learning. What does it really refer to? In order to make it a bit more concrete, a lot more concrete, researchers in cognitive neuroscience or in engineering and AI have built algorithms that are there to emulate a particular function, like learning. They are learning algorithms. They are algorithms of different ideas, like attention is algorithms for attention, is algorithms for motor control, and things like this. So we've been interested in relating our understanding of neurons, dendrites, synapses that have been built in a computational model, in the mathematical model, with the algorithms of learning. So by connecting the math of the physical system with the math of the algorithm, it's a bit easier because they both are in the same language. It's a bit easier to see which one connect with which. And then if we have that connection, we effectively have made a connection from neural data to computational model to algorithms to a function. So that's the 4-step approach.
So the modeling is a great part of it. How can you understand if a model, it goes back to the the part that you were speaking about, a physicist might try to oversimplify because a physicist wants to make the model. So how can you assess if a model is actually working? Is it representing what you want it to represent,
Right.
or is it like overlooking in some parts?
Right. So what's important here is to really have a closed loop with data. So data really has to be closely involved in the shaping of the model itself. Actually, you have two questions in this. It's like, how does it oversimplify? There's two aspects of simplification. One is you can oversimplify in the sense that you can lose accuracy. So you're getting to be wrong about something. And that's something you can tell by looping in the data. If you predict something, then you test it with the data and you don't get it, your model is wrong about something. You don't have good accuracy. So that's one way to be wrong. But the other way to be wrong is a question of precision. So you can build different types of models. Some are more simplified than others. And in terms of the level of granularity that they hold. So we call that levels of description. So you can, have a model that has an entity in there that's a dendrite. It's a pretty small thing. But there's other models that's just modeling a part of the brain that's being active or not, where a dendrite doesn't really exist. So the model that tackles a part of the brain is not very precise. It's good to capture fMRI data, but it's not good to capture dendrite data. So that's where you can oversimplify. So you have to adjust the level of description to the task that you're trying to solve. And that's a very hard thing. And it's something I try to teach students when I'm teaching computational neuroscience, is how do you choose the right level of description depending on the type of challenge that you're facing?
That is very cool. I'm sure physics comes handy when you're trying to do that. I'm just wondering, you mentioned that you're doing some work for neurochemicals, especially serotonin. I myself been really interested into the whole idea of neurochemicals and how brain is trying to like basically have that kind of balance. I'm just wondering if you can explain more a little bit more about the serotonin and what is it that you're actually working on in your lab.
Yeah, that's very exciting. Right now, we're working on a manuscript that I think will have big impact on the theory of serotonin. And if I have to reuse that four-step program that I'm referred to, I worked with actually the student that just graduated that I referred to earlier, the PhD student. He started doing a master's in a neuroscience lab. So he was starting with experiments where he was recording from serotonergic neurons in the part of the brain that releases serotonin. And so that's the sort of fundamental data that we use to build a model of the serotonergic system. We realized that the serotonin neurons are having a particularly strong adaptation. So they when you force them to fire, they respond, but then they adapt very quickly and they don't respond very much. And there's this sort of surprise type of response that they give naturally just due to the mechanisms are there. But that's okay. Then we were wondering, okay, how can we connect that to some type of function? Now, in the field of serotonin, people have suggested many types of function, and there hasn't been a consensus. And there's been a lot of contradictory evidence. One line of research has shown that serotonin neurons tend to respond when there's a reward. So if you give a reward unexpectedly to an animal or to a human, serotonin neurons start to respond. But in a way that's very puzzling, if you give a reward that's expected, serotonin neurons stop firing. And in a way that's even more surprising, if you give a punishment that's unexpected, serotonin neurons respond too. Oh, Siri wants to talk. So this gives us a puzzling set of observations. Serotonin neurons respond to unexpected reward, but not to expected rewards, and they respond to punishment. How can that make sense? People said, oh, serotonin neurons just respond to surprising things, but this didn't capture the fact that on average, if you're like in a rewarding context, the baseline activity of serotonin is increasing. And if you're in a more punishing context, the baseline activity of serotonin neurons is decreasing. So like, how can we capture all of this together? And what we did is we went to the algorithm level. And we look in reinforcement learning, that's been a world, part of artificial intelligence, that's been very conductive in letting artificial agents navigate complex environments. One quantity of importance there is the idea of a value function. Now, a value function is meant to represent how much reward is the animal expecting to get, or the agent is expecting to get in the near future, given its current state. So it's like the prospective aspect of how much reward you would get. This depends on a lot of things. It depends on the context. It depends on what the animal has seen in the past, and so on. And we combined that was able to explain why unexpected reward was making neurons firing, because if you have an unexpected reward, you're like, oh, this is valuable, it's very important. So you get reward, and you get a reward signal. It also explained why if you expect a reward, there should be a decrease in the serotonin, because as you get the reward, you're not likely to get another one in the future in most of the tasks. And therefore, your expectation of getting a reward in the near future drops as soon as you get the reward. So it explains that. And what it doesn't explain is why punishment gives neurons to increase their response. And that has to do, we think, with the adaptation. So the fact that the neurons are strongly adapting. So when you give a punishment, there is actually a negative response in the neurons that we tend to not see given the way we record for neurons. And then when the end of the punishment happens, the neurons start to respond because they were adapted and there's an overshoot. And that fits with what we see in the neural recordings. So all that to say, we think that the neurons are encoding, the serotonin neurons are communicating to the brain the expected reward in the near future.
Right. Yeah. Serotonin is one of those really amazing molecules. I've been just reading some papers about psychedelics because psychedelics have a huge effects on releasing serotonin, and I'm kind of interested into the whole spirituality of the things that would cause by releasing serotonin. And I'm sure there's been like really cool researches, and maybe that would be something that I really want to like explore later on in my life. But yeah, it is a cool molecule.
Yeah, and the psychedelics are affecting the receptors of serotonin mostly, and that's something we don't know. It's something like we have one step of understanding now. We feel we understand why the serotonin neurons are firing, but what we don't know is what this does on the postsynaptic side. So when you send serotonin, what it does to the system, we don't know what it does. There's as much ambiguity there as to what exactly it does. We know things about psychedelics, and this is also very puzzling for many reasons.
Yeah.
So I was wondering because as you were also describing your current focus in research, you like specifically referred to neural dynamics. So if we shift back a bit in your career about neural dynamics, what happens? Now I'm just reading exactly like the introduction paragraph to this. What happens in our brain when we make a decision? So I was wondering if you could speak a bit about that, about neural dynamics in general, and how you use that right now in your current focus of research. How does that help you?
Mm-hmm. Yeah, so what happens in our brain when we make a decision has to do a lot with what we call the neural codes. It's like we know that when we make a decision, there's some elements of the brain that gets activated and they represent the choice that we've made. And then if there was some ambiguity in our choice, there was some fighting between different parts of the brain that were fighting against another to make that decision. Now, what are those parts of the brain, and how can we find them in the brain exactly? And that's the problem of neural coding. How is that information represented in the brain? And I think, in my mind, there are two lessons about neural coding that are important, at least in cortex. Now, well, maybe three. The first lesson is that it really depends on the part of the brain you're dealing with. Different parts of the brain have vastly different approaches to encoding this information and encode or mediating this competition that I was referring to earlier. And I like this idea that if there's multiple ways of doing one thing, the brain will have used all of them. It's not as if the brain will say, you're like, oh, why did the brain not invent the wheel, or like, why are the planes always using the same type of wings? We have a tendency when you're building things to build things the same way, but biology doesn't work this way. If there's multiple ways of doing getting to a goal, it will use all of them, and often at the same time. So there's a lot of diversity in neural coding. Then the other lesson is that of the concept of rate. So neurons represent information with the fraction of neurons are responding at within a small period of time, like within say 100 milliseconds. The fraction of neuron responding within 10 milliseconds is representing something. Like if there's for instance, if you have a more light intensity in one part of a visual field, the ganglion cells in the retina will fire more action potentials per unit time. The time at which, and the amount of action potentials will be proportional to the intensity of that pixel. But the other element is, the third element is the idea of distributed coding. It's not as though there's one neuron that's responsible for representing one thing. Anything is represented by multiple neurons in a distributed fashion. Some neurons are more associated with that and others are less associated with that. And it's by working as a team that they represent that information. Now it becomes really complex to navigate because this distribution is overlapping with other concepts. So every neuron is actually coding for a little bit of everything. They're not coding
Yeah.
for one thing. It's only by looking at the population that you can disambiguate this. This is, by the way, a place where computational tools have a very important role to play.
Mhm. I'm just wondering, so you mentioned earlier a bit of like what kind of techniques you use to measure those kind of activities, but back to serotonin again, how is it that you actually measure the effects of those channels? Is it like fMRI, EEG, MEG?
Yeah, yeah. So this is an important question. Like, you can address this with different levels of description. I am dealing with animal studies because I want to get to a more precise, almost like gene level description of uh, how serotonin or any sort of computational framework applies, which is really hard to get in humans, where you can't know with different cell types, you can't, precisely record from a part of a cell, let alone a cell type and so on. And so, yeah, I deal mostly with animal studies. I read also the human or primate literature, but I will mostly focus on animal studies where you can, record from specific cell type in a specific part of the brain and get like single unit recordings. You can identify one neuron from another, something you cannot do in fMRI. And then you can, selectively block or excite this particular cell, or you can selectively block or excite this particular pathway. So this allows us to get that level of granularity. I'm not interested in the level below either. People are, and it's important. There are molecules that are involved in all of this. There's GPCRs. There's a lot of different types of molecular engineering that the brain has developed. That's very interesting and very important. I'm not getting there because if you start to get that level of granularity, you can't get close to the whole brain, right? Like, it's so small that you have to understand just a subpart of the brain. So it's a choice. That's the level of description that I like.
So you also mentioned genes. I was wondering, are you also, well, do you take the is it also part of your research, like the different serotonin receptors and maybe their regulation? Maybe how is the gene expression of each and every different one of these serotonin receptors, that of course they all do something different to make the life of students more difficult, but what role does that play in this equation?
Yeah, so this is something that for serotonin in particular is puzzling because of the serotonin receptors have the largest diversity of receptors. Like, they're so diverse, so many different types of receptors, and they have so many different types of postsynaptic effects. And yet, like, we don't know the real effect that each of those receptors have on the system. We so this is a very puzzling thing. I haven't in like This will be a target for future research that we would like to tackle. We haven't been able to make any significant progress in that direction. We are talking about it very often, but we right now don't know exactly how to address this. One approach is an interesting one, is the approach of psychedelics, because psychedelics have a huge effect on the system, and they target a subtype of receptors. And so at least you have an effect on the system that you can start to study. However, psychedelics tend to like have a crazy effect on the system, and it's not not to play on words very much, but it's like it saturates the receptors typically to an extent that you're wondering whether you're just not triggering a non-physiological regime, and you're wondering whether it's really relevant to understanding the serotonin signaling to study psychedelics. But yeah, right now that's one of the meaningful pathways to discovery that we think is happening.
That is very cool. I'm sure there's still a lot of unknown things about serotonin, and maybe us as a students can go and definitely explore that. Let me just shift the gears a little bit now and talk about the stuff you do other than neuroscience and kind of your hobbies. What do you do outside of university and your personal life that
Hmm.
you enjoy?
Well, I spend a lot of time with my family, but as a parent, and of course that's a lot of time consuming. But I'm also a big fan of music. I like to listen to music mostly. I do play music, but not very much. I like to listen to music quite a bit, and I will travel to go to different concerts. I have a very wide taste in music. And then otherwise, I like, physical activities. I would cycle a lot. I do climbing. I do running. I'm a runner. It keeps my. This is actually an important aspect. Everything is connected in this, right? Like I find that the most creative time I have is when I'm running. So most of my most significant ideas that led to important papers were done running. So I went for a long run, and at the beginning, like my mind's just clogged, and after maybe like 5, 10 kilometers, then the mind's kind of opening up, and there's a more creative type of association that takes place. So it's an
Cool.
important aspect of my life.
Yeah, I'm sure.
Could you also share with us the experiences you've had with failure during your academic journey or just in general? And how does that help you, or if even might have not, but how does that affect
Mhm.
the way that you approached?
Well, for one thing, like uh, failure is an integral part of what we do, right? Like you always feel like you're going to fail the next step. I always feel like I'm going to fail the next step. I feel like while I have a good trust in what I do, I still have a lot of criticism about everything I've done, and therefore both sides. I feel like that's what a scientist should have, like seeing always both sides of the equations, um, and see the strengths and weaknesses in everything you do. Not thinking that everything you do is just so amazing all the time. Like that there are limits to everything you've done. You try the best all the time, of course, but it never works splendidly. Um, so there are two things I can mention to this. One is I don't know if I would call that failure, but um, in I remember during my PhD, the sort of failure rate might have been higher than it is now, and there's certainly a good degree of persistence that had to happen. I What I remember from my PhD is I would cycle to work, have an idea when I was cycling to work, get to the lab, get to work on that idea. Like, oh, I'm so excited! It's so cool! And then my lab members would come in the lab, and I would tell them about my idea. Not my lab. I mean the other colleagues, like other PhD students. And then they were like, "Oh yeah, that's cool, but did you think about that?" And oh no, oh, and then oh yeah, but that's not too bad because that. Oh no, you're right. And then discuss the idea, and then by the end of the day, you're like, "Oh no, it's a bad idea. Forget it. Done." And then the next day would start. It would be very similar. So, you'd have to go through multiple ideas like this, and in retrospect, many of my ideas were very ridiculous, like were very simple, and I would not even consider those ideas now. But mainly because I've been through this, right? I had to fail like 300 days a year in order to get to a good idea in the end, one that would actually sustain or withstand scrutiny. So that's one sort of failure I would have. And then otherwise, there's just so many other types of failure that I could think of, like papers that didn't go through, grants I didn't get, um, that I felt like I would be deserving for various reasons. And it's important to not think about this too much. You shouldn't celebrate too much your successes, and you shouldn't uh, cry too much about the failures, and just keep on going to the next thing. It's hard for one day, and then the next day you can rationalize things and go to
Well, I
go to
guess
the
just
next
like
step.
a serotonin receptor, punishment comes with adaptation. So
That's
you
right.
can think of
That's
it that
right.
way.
That's right. Yeah.
So I just wanted to ask, because now you mentioned about having different backgrounds, about different backgrounds of students in your lab, and how each and every one of them can be a source of weakness or strength, and how that can just open a new whole vision. I was wondering if you would talk about how a student with biomedical background can approach your work. What are the things that they should be mindful of? What are the things that they can bring to the table?
Especially tips
That's
about like how to get involved more in physics and math aspects,
right. Right. So I think, yeah, I mentioned how it is once you get into my lab. I think like I typically give the students a few critical courses that they would need to ideally get, and then everything else. Because of course, when you start grad studies, you are at the transition between learning with courses and learning independently. So there's a few places where it's important to learn with courses because you do some boring math, but it's important to get your mind wrapped around this that you wouldn't do otherwise. But after that, you learn by yourself. Um, but before that, so before getting into my lab, that's a question I get very often. And what are the skills? Because there's a lot of people in biomedical studies that actually have a very strong interest in computational aspects. Some of them are mostly interested in coding, and there is not that many coding courses out there for you. I'm trying to set up the right one. I mean, I think in TMM, so you have one now. It's R. Now we're trying to make a second one for Python. So they have a few courses in coding. If you can take those, and it's typically adapted to your particular stream, it's is very beneficial for sure. The other aspect is like probability and statistics. Um, and that you typically have a little bit, but not to a very far extent. Um, Probability is a very important aspect that I tend to suggest to the students. And the third one is that I think is nice to if you can choose an elective in that direction, it's very useful. Is linear algebra. Linear algebra, vector spaces, this type of concept is the basis to a lot of machine learning. And without linear algebra, it's really hard to get into this. And it's also the basis of a lot of concepts that are not in machine learning, more like in computational simulations. There's another one that I don't suggest all the time. It really depends on what the student wants to do, is differential equations. Differential equations, systems of differential equations, something physicists study very much. This is a mixed bag. So it really depends on if you want to go in that direction or not. Simulating systems that are dynamical requires you to understand differential equations, but you don't necessarily have to take a differential equation course to see all the different types of differential equation, when in computational neuroscience, we only use one. So this is, if you happen to stumble on this, like this, go ahead, but it's not the first thing to do.
I would also like to ask about what are the different, like general differences between a lab that does more theoretical research and a one that does more experimental one. So,
yeah, I like that question. Yeah, it's quite different for many reasons. Of course, like we don't have to be at the bench all the time, and there's a different rhythm to things in the sense that there's an important creative aspect, right? You have to come up with an original idea. And so in order to come up with these original ideas, it's important to be in a state where you can, be creative. But really, that happens only after you've understood enough of the field. It's hard to really be creative if you don't know anything. So you first have to learn quite a lot of things. There's a lot of learning going on about, everything we just mentioned in particular. And after that, there's also a certain level of industriousness that you also have to apply. So people might have a preconception that theorists are just there dreaming, but no, you do have to get to be efficient at learning new paradigms and be comfortable with them and applying new paradigms to a particular project. In my lab, I typically give people that come in the lab, call that the baby project, but it doesn't mean the project is easy. It's the project that you start with that I have figured out. I have understood where this should go, where it ends. I have made typically some of the main ideas, and I know it can get to a paper within roughly a year. It can be 2 years, and this allows students to sink their teeth into something, and they learn a lot into this. There's not much creativity though, and so they have to be industrious, just as an experimental person has to be industrious. It's just they don't have to be in the lab all the time. They can be at home, they can be in a cafe, it doesn't matter. But there's an element of productivity just as much. And then after that, there comes the creative phase. So the second project in a PhD, if it's a master's, that's all there is. But if it's a PhD, then the second project is a discussion between me and the student. We're talking about different ideas, and we come to the project together. We come to the gem of the project. And typically there's a third project where the student's completely independent, came with the idea on themselves. If we reach that point, I feel like I've accomplished something. I feel like I've trained someone to become a scientist that can come up with their own project, and I'm very proud of this.
That's
really
great.
Exciting to get to that level.
Yeah.
I'm just wondering, so you mentioned a lot of things about your journey, your failures and stuff. If you go back to the ages of your undergrad, what would you tell yourself, like a piece of advice, or like any other undergrad students who are interested in doing either research or get involved in neuroscience? What would be that key thing that they always have to keep in mind?
Right. Yeah, one thing, it's something that maybe I knew, or I don't know. I didn't know how bad I was at communication before getting to where I am now. It's something maybe physicists do differently, because in physics, there's, at least in the training I've had, there was very little emphasis on giving talks or making nice written presentations. It's not the case in biomedical sciences. I find people in TMM or in biomedical program tend to have more experience in writing reports, maybe to the point that it's too much. But I realize how, like, it's something that wasn't clear to me at the beginning, that being an academic is not just about research. It's not just about understanding new concepts. It's also a lot about communication, and communicating the new concept that you've discovered, or just the things that you've learned are not necessarily new, but that you've learned. Communicate that to other people. When it is, when you're writing a grant, which is an essential step, you have to communicate why the research you propose is very, very important. That's a communication skill. And I guess at the very beginning, I didn't think that this was important. And I see students coming in my lab and they think they can succeed without being excellent communicators. I'd say this, but at the same time, I also see people that succeed while being excellent communicators and very bad scientists. So I'm saying to myself, you are maybe a bit silly to think that you can succeed without being an excellent communicator. But please keep on thinking that science is the most important thing, right? Like getting to an accurate science remains the most important thing, but I was a bit naive in thinking that I could dismiss the communication. Um, yeah,
That is
but
very
I'm glad
interesting.
that
Yeah.
I remained focused on the science because otherwise I might have become a bad scientist.
So
Hopefully,
how do you think we
I
can
haven't
like,
become one. I don't know.
mhm, right. How do you think, like, is it by just giving more talks or giving more reports? How is the way that we can improve that communication skills in the most efficient
Right.
way?
I think you've said the main thing though. Like giving more talks is certainly one of the best way to get better. Um, I used to be frozen in front of people, and now if I have a little bit of a gap in my sort of agenda of giving talks, I see that I will start to be stressed, and that the quality of my talk will decrease. But if I keep on talking in public, it just stays fairly good. But you also like in the process, you have to be mindful of what you're doing. You think an important thing is when you listen to a talk and you think that's good, ask yourself why is it that you find that's good. Same for writing. When you write a book, it doesn't have to be science, and you're like, wow, that's well written. Why is it? What is it that they do that you think is working, right? And by constantly asking that question, you learn bits and pieces and you improve.
What kind of positions do you have in your lab that are available for undergrad and maybe other students, like either they're applying for honors or even grad school? Are you taking volunteers or are you taking honor students? Just maybe give us a introduction about like what kind of opportunities you offer currently in your lab.
Well, I don't take volunteers because we're not allowed to somehow. It's a crazy setup, but um, um, I used to, but I don't anymore. Undergrads, they are always welcome in the lab. I see this as a learning experience. It's really for the student to get an exposure to grad studies. But it's done through NSERC summer research programs or Europe, so the undergraduate research. At least I think this is also gone, right? So it's mostly NSERC summer research or winter research programs. I've benefited from this as an undergrad very much, and I feel like it's my duty to give forward in this time. I remember being in meteorology and learning how to code them on a big like atmospheric science model, but I didn't benefit anything to their project. I just learned coding. So that's the sort of position that is in mind for that. Then masters and PhD, that depends on the state of the lab. There's some like the best way is to contact me a year ahead of time, so that I can have time to plan for space. Typically, if you ask me for now, and there's no space, my lab's full, but maybe in a year from now, it would be. Yeah, and
Yeah.
I'm interested in students that are really interested in the type of thing we do. We're not just doing neuroscience. Maybe 99% of the emails I get are people with a pharmaceutical background, and they haven't really read what I'm doing. They cannot really be part of my lab. So, yeah.
Perfect. Well, there you go, folks. You heard it from Dr. Naud himself. So try to really get on those communication skills, math and physics, and be able to continue there. Awesome. Thank you very much, Dr. Naud. It was such a
Thank you.
pleasure to talk with you today, and hope you have a good day.
Awesome. Thank you. Dr. Naud, we'd like to thank you again for accepting our invitation and becoming a guest of our podcast. I'm sure students and other people listening to this will have great ideas of how to continue their interest in not just neuroscience, but in just becoming a well-sphered student, let's say, in science and try to approach other parts of it. Yeah, thank you.
Thank you for listening to this conversation with Dr. Naud. In this episode, we explored his inspiring journey and exciting research happening in his lab, including dendrites, serotonergic neurons, and biophysics of microstimulation. Dr. Naud also shared valuable advice on decision making, turning failure into growth, and preparing for possible opportunities in his lab. Lastly, he highlighted the importance of building strong communication skills to succeed in science and life in general.
This is a Dendrite podcast. To support it, share this episode with your friends and anyone else who might find it interesting. And be on the lookout for our monthly journal club meeting. Thank you for listening to us, and we hope to see you next time.